Hello and welcome to our sponsored content today. My name is Martin Mangold and I'm an application specialist at BMG LabTech. Today I would like to introduce the CLARIOstar Plus microplate reader, which plays an important role in the research Professor Morgan will present later on. I would like to highlight which aspects make it the best tool for the measurement of real-time cell-based redox assays. So, to give you a quick introduction of BMG LabTech, we are a specialized microplate reader company from Germany with more than 30 years experience in the field. BMG LabTech offers a range of single-mode to multi-mode readers with industry-leading specifications, helping our users to accomplish all aspects of research, from basic research to high-throughput screenings. The CLARIOstar Plus is one of the most advanced readers we have within our range. This multi-mode multi-mel instrument can accomplish all of the technologies you see on the screen, from fluorescence intensity to alpha screen measurements. Users can specify to include the technologies important to them upon buying this instrument. Its standard functions include the capability to read all microplate formats from 6 to 384 well plates and can be used for both endpoint and kinetic detection. It allows for sequential multi-excitation and emission, multiplexing and ratiometric measurements, which are especially useful for the analysis of conditional signal output, such as, for example, by redox-sensitive probes, which change the excitation properties depending on oxygen levels. So, why do we consider the CLARIOstar Plus as the ideal instrument for measuring real-time cell-based redox assays? First off, it keeps your cells happy. The optional atmospheric control unit, or ACU, can maintain a physiological environment for your live cell assays. With the regulation of carbon dioxide and oxygen levels from 0.1 to 20% and a gas ramping function, the ACU can induce hypoxia or reoxygenation. In conjunction with the built-in temperature regulation, this enables the real-time assessment of oxidation states in cells, cell proliferation or cell viability under various atmospheric conditions. Second, the CLARIOstar Plus produces high-quality data and sensitivity. This is in part achieved by the linear variable filter monochromator, which offers adjustable wavelengths and bandwidths and is the most sensitive monochromator on the market. The CLARIOstar Plus can record excitation and emission scans, and filters and monochromator can be combined for further flexibility. Another advancement with the CLARIOstar Plus is the use of the capability that we call enhanced dynamic range. This feature expands the luminescence and fluorescence detection range to 8 decades in one reading. For further ease of use, it automatically identifies optimal settings for your assays with no need for manual optimization, which is especially interesting for experiments with increasing signals over time, such as kinetic assays. Another key to high-quality data collection is to be able to focus in on the appropriate place in the well to collect your data. The automatic focal height adjustment of the CLARIOstar Plus allows you to focus with a 0.1 mm resolution, improving sensitivity for top and bottom reading. In particular, the bottom read performance is beneficial for the focus on adherent cells or cell layers. Since adherent cells are not always confluent and signal distribution can vary throughout the cell, our well scanning options can really improve data quality for such heterogeneous samples. Scanning options range from orbital well averaging and spiral well averaging to matrix scan with up to 900 data points per well, allowing you to localize your cell populations. with up to 2 onboard reagent injectors with adjustable injection volumes per well and the option for multiple injections per well, reactions can be started or stopped at specific time points. Due to simultaneous injection and data collection, you are able to measure cellular events in real time. All these features enable us to deliver on high-quality results for users of our instruments. Finally, we provide useful data analysis tools with our microplate analysis and reduction software MAS, which is included with every reader. This multi-user platform allows you to view and analyze your data and offers a plethora of capabilities for data analysis, from kinetic analysis, standard curves, statistical analysis, to ratiometric calculations, and much more. So, to summarize, the CLARIOstar Plus with ACU is the perfect platform for your cell-based redox measurements. It keeps your cells happy and provides valuable features for the adjustment of oxygen levels. It produces data of the highest quality and provides data analysis tools for these data in the form of MAS. At this stage, I'm going to finish my presentation. Thank you for listening and for your attention. Now I would like to pass on to Professor Morgan to deliver on the main part of our webinar today, which will deal with the monitoring of redox enzyme activity in living cells. Thank you very much.
So, hello, my name is Bruce Morgan, and I'd like to welcome you to my talk, where today I'd like to tell you a little bit about our work in which we're using redox-sensitive green fluorescent proteins, roGFP2 specifically, roGFP-based approaches to monitor redox enzyme activity inside living cells. So I'm sure many of you are familiar with roGFPs and the probes that are based upon roGFP, but I just wanted to briefly introduce the concept here again. So what you see here is a genetic fusion between roGFP here in green and a glutaredoxin here depicted in red. And in the case of this probe, what will happen is that the glutaredoxin will facilitate the thermodynamic equilibrium between the thiosulfide redox couple on the roGFP and the 2-GSH-GSSG redox couple of the glutathione pool. And what this means is that if we know the redox state of the roGFP, we know automatically the redox potential of the glutathione redox couple. And we can monitor roGFP oxidation inside living cells very easily by monitoring this ratiometric change in the fluorescence excitation spectra. And a lot of our work in the past has involved using these roGFP GRX1 sensors to learn more about glutathione redox homeostasis inside living cells, and in our case, specifically in the context of yeast cells. And from both our own work and work of that of a number of collaborators, what we've been able to show is that the cytosolic glutathione pool is actually extremely reduced and extremely robustly regulated. And we're talking about a glutathione redox potential somewhere in the range of minus 320 millivolts here, which roughly equates to a GSH-GSSG ratio in the range of 50,000 to 1. We were also able to show, together with Jan Riemer in Kaiserslautern at the time and Maya Schurdinger at the Weizmann Institute, that mitochondrial peroxisomal glutathione pools are also similarly highly reduced. In the specific case of yeast, we could show that one of the reasons that the cytosolic pool looked so different to what we might have expected on the basis of classical whole cell analyses of GSH and GSSG content is because the GSSG within the cell is highly compartmentalized. And in the case of yeast, we were able to show that the ABC transporter, YCF1, efficiently transports all cytosolic GSSG that's not immediately reduced by glutathione reductase, for example. It will transport this GSSG to the vacuole where it will be stored. And we're also able to show that oxidative stress-dependent changes in cellular GSSG levels are completely abolished in strains deleted for YCF1. In other words, there's an enormous capacity for GSSG reduction, and if we remove the ability of the cell to compartmentalize GSSG, then our whole cell readouts look much more similar to our cytosol-specific probe-based readouts. So this is a summary of the work that we've done so far, looking specifically at glutathione redox homeostasis. And essentially what we're doing here, as I've already mentioned, is using the change in roGFP oxidation to tell us something about a specific metabolite of interest. In this case, the glutathione redox couple, but we can also develop probes based on roGFP, for example, for monitoring qualitative changes in hydrogen peroxide. And so it's always the case that we're interested in monitoring the redox species here. But we wondered whether it would actually also be possible to use roGFP readouts to monitor not redox metabolites, but actually the activity of the enzyme that facilitates the equilibration between the redox metabolite and the roGFP redox state. And so kind of loosely you can think of this as asking, as the difference between thermodynamics, where we're always interested in what is the roGFP redox state at any one time, and what does this tell us about the redox state of the redox species that we're monitoring, to a question of kinetics, where we're asking if we change a redox metabolite, the level of hydrogen peroxide, for example, how quickly does our roGFP equilibrate with that change? And we know that the speed at which this takes place is dependent upon the activity of the enzyme facilitating this equilibration. So we've already shown before, together with our collaboration partner Marcel de Ponte, that actually the transfer of oxidation from the redox enzyme, for example, peroxyredoxin or a glutaredoxin, to the roGFP is actually not rate limiting for the roGFP oxidation. What's rate limiting is actually the oxidation of the redox enzyme itself. And so we asked whether we can monitor the rate of roGFP oxidation, and from this learn something about the activity of the redox enzyme that's facilitating this equilibration. So, as a proof of principle, we first turned to a glutaredoxin, GRX7, from budding yeast. And the reason we chose GRX7 is because Marcel de Ponte, our collaboration partner, had already extensively characterized this in vitro. The other reason was that it only contains one cysteine residue, so this greatly simplifies the number of possible, the possible redox chemistry that can take place. And what we did was to express a fusion construct between yeast GRX7 and roGFP in yeast, where we're hoping to use yeast essentially as a cellular test tube for rapid screening of different constructs. And we induce oxidation of the glutathione pool in the yeast by addition of hydrogen peroxide, and then we rely upon the production of glutathione disulfide, which will lead to the oxidation of glutaredoxin, and this oxidation is transferred onto the roGFP, where we can read out the change in roGFP oxidation. To do this, we have a semi-high throughput method based upon a 9612 fluorescent plate reader, so we can monitor multiple conditions in parallel, always on the basis, as I said, of the fluorescence excitation spectral changes. And here we have several of these BMG LabTech CLARIOstars in our lab, which we have used for many years now, and they produce a readout something like this, where you can see multiple different wells with different samples and different conditions, and we can very quickly screen through lots of mutants and lots of conditions in this way. So the first thing that we had to do was establish a yeast system that we could use to actually enable us to address these kind of questions. And the first thing that you notice if you express these constructs in wild-type cells, so on the left panel here we have an unfused roGFP as a control. In the middle we have our GRX7 roGFP fusion, and on the right-hand side the fusion in which the active cysteine residue has been mutated to a serine. And you see that there is some response of the GRX7 roGFP fusion, but it's not that great, to be honest, and it would be difficult to interpret anything from these responses. So the first thing we did was to try to sensitize the response to hydrogen peroxide. So the first thing that we did was to delete glutathione reductase, and now we find that the glutathione pool is much more responsive to H2O2-induced oxidation. But now when you compare the three constructs, again you don't see very much difference. You see that in the case of the GRX7 fusion, the roGFP oxidation is more rapid, but again it's very difficult to draw meaningful conclusions about the activity of GRX7 based upon these differences. And the reason is we know because the roGFP is also very efficiently oxidized by the endogenous glutaredoxins in the cytosol of yeast. And so as the next step, what we did was to create a triple mutant yeast strain, deleted for glutathione reductase, as well as the two cytosolic dithylglutaredoxins, GRX1 and GRX2. And now you see a system in which we can, in which the roGFP oxidation is very specific, dependent upon the activity of the fused glutaredoxin. So you see that our unfused roGFP here barely responds at all to hydrogen peroxide, as is the case with our cysteine mutant GRX7. We only see this efficient and sensitive and specific oxidation of roGFP in the context of the GRX7 fusion. So we now felt confident that we had a system, a yeast system that we can use as a cellular test tube, as I said, to screen, to understand better the mechanistic basis of GRX7 activity. So how do we interpret the data? So what we decided to do was simply to take the area under the curve of roGFP oxidation for the first 50 seconds of the reaction and to plot this area under the curve against the H2O2 concentration. Okay, so we then asked whether we can use these assays to do some meaningful protein biochemistry. And the thing that really interested us was to understand what are the defining structural differences between enzymatically active glutaredoxins and enzymatically inactive glutaredoxins, for example GRX5 from humans, which we know is important for iron sulfur cluster biogenesis. So the first thing we did was to test fusions of GRX7 and GRX5 to roGFP. As you've already seen, the GRX7 roGFP fusion responds nicely to hydrogen peroxide, whereas with the GRX5 roGFP fusion, we see barely any response at all. So we now asked if we could use this yeast assay to start screening different mutants of GRX7 and GRX5, and in that way to gain some insight into what were actually the determinant structural features of enzyme activity or inactivity in glutaredoxins. So you may notice that there are actually many differences that you can pick out between these, conserved differences, I should say, between enzymatically active and inactive glutaredoxins. And some of the most important ones are highlighted here in these diagrams. So you see, for example, here is the redox active cysteine. You see that within the active site, in the redox active glutaredoxins, you typically have cysteine, proline, tyrosine, whereas almost universally in the redox inactive glutaredoxins, you would typically find cysteine, glycine, and phenylalanine. Another difference around the active site is that immediately preceding the active cysteine, in the active GRXs, you have this very short loop structure, whereas in the redox inactive glutaredoxins, this loop structure is greatly elongated, which may change subtly the positioning of this redox active, or not redox active in the case of the inactive glutaredoxins, would change the position of this cysteine residue. And a third important difference is you see here, whereas we often have charged residues here in the redox active glutaredoxins, in the redox inactive glutaredoxins, these charged residues, positively charged residues, are often replaced by bulky hydrophobic residues such as tryptophan and or by a proline residue. And so we asked whether these motifs in particular were important determinants of enzymatic activity. So the first thing we did as a proof of principle was just, for example, to try what happens if we mutate this tyrosine here to a phenylalanine. So surprisingly, actually, when we mutated the redox active tyrosine to the phenylalanine that we typically find in the redox inactive glutaredoxins, we actually saw an increase in the rate of roGFP oxidation, which was somewhat of a surprise. When we mutated this tyrosine to a histidine so that we still have some kind of aromatic side chain, but of course it's now quite a different amino acid, we still retain quite some activity. When we mutate this tyrosine to an alanine, we find that the activity now is largely abolished. And importantly, when we plotted the area under the curve of these roGFP responses, we saw that the pattern very nicely paralleled the pattern that we could obtain via classical in vitro characterization of the enzymatic activity of these mutants, either against GSS-cis here or against GSH. And we saw this high degree of consistency between these in situ roGFP responses and classical in vitro measurements for more than 10 other tested residues. So this was an important kind of, for us, proof of principle, and then gave us the confidence from this point on to proceed with the roGFP only measurements. So I just wanted to make an important point of clarification here. We're not saying that these roGFP measurements are quantitative. They're definitely not quantitative in the same way that classical in vitro characterization is, but we believe they're semi-quantitative. You can clearly see differences in activity which closely parallel differences that we can measure in vitro. And what we really want to try to say, or what we're really saying, is that we can use these yeast-based, roGFP-based assays to rapidly screen through many, many mutants, much more quickly than we could do using classical in vitro approaches. And when we find interesting differences, we can then follow up on this, of course, more specifically in vitro. Okay, so having seen this consistency between the roGFP and the in vitro measurements, as I said, we now had the confidence to proceed with roGFP-based, roGFP-only measurements in order to start trying to better understand what really are the determinant differences between redox-active and redox-inactive glutaredoxins. And again, I come back to these diagrams, and you see that there are, as we've already pointed out, several places in which we have these conserved differences between the active and inactive glutaredoxins. And so we started to make constructs in which we tried to turn the human GRX5 progressively, motif by motif, into more and more of a GRX7 or a redox-active like glutaredoxin. And to summarize a lot of data, what we actually found was that there was not one single structural feature that determines whether a glutaredoxin is enzymatically active or not, but actually it's determined by all of these different structural features in combination. So you see here, in this H2O2 dependent manner, we have an increase in the rate of oxidation as reported by the area under the curve of roGFP oxidation, driven by GRX5. You see for the wild type GRX5, this is negligible. There's a negligible roGFP oxidation. But as we progressively, one by one, change the different motifs on GRX5 to become a redox-active glutaredoxin-like motifs, for example here by exchanging the loop, or by changing the active site glycine to a proline, or by switching these positively charged residues, or introducing these positively charged residues in place of the tryptophan and proline motif, and so on, we progressively, step by step, increase the activity of GRX5 until when we've substituted different combinations of 3 of these motifs in parallel, we have an enzyme that's approaching now the activity of GRX7. So we almost completely converted a redox-active glutaredoxin into a redox-active glutaredoxin. The take-home message is that several different structural features in combination are important for determining whether a glutaredoxin will be redox active or be redox inactive. Okay, so these kind of approaches are not actually limited to glutaredoxins. We recently published a paper also where we're using similar roGFP based approaches to characterize peroxyredoxins. I don't want to go into too much details on that today for time reasons. What I did instead want to do was to introduce another assay that we've developed recently, a new approach, which we think can even better enable us to characterize redox enzyme activity. And the assays that we have so far work pretty well, I hope, as I hope I've been able to convince you. But there are question marks, there are unknowns. And one of the unknowns is that when we add the hydrogen peroxide, we don't really understand in yeast, surprisingly, exactly how hydrogen peroxide leads to the oxidation of glutathione to glutathione disulfide. But we have some reason to believe that this is in part dependent upon peroxyredoxins and or glutaredoxins. And of course this then complicates things because maybe the glutaredoxin that we fuse to our roGFP might also be important for producing the GSSG in the first place that will lead to the oxidation of roGFP. And so we asked if it's possible to develop a more direct assay where we can directly apply GSSG to our probe-expressing yeast cells. And to this end, we worked with OPT1 overexpression. So OPT1 is a known plasma membrane localized reduced and oxidized glutathione transporter. And we asked if we could overexpress this in our triple mutant cells, so our cells deleted for glutathione reductase, plus the 2 cytosolic dithiogluteredoxins, and use this to titrate GSSG into our cells to lead to the oxidation of our roGFP GRX fusions. And I just want to show you a couple of data slides on this, and I can show that it's nicely the case. And actually, when we do this, we get a much finer resolution, a much more linear change in the rate of oxidation and the degree of oxidation of our roGFP constructs than we can achieve with hydrogen peroxide addition. So you see here on the top, these are empty vector expressing triple mutant cells. When we apply H2O2, as we've already seen, we have this increase in roGFP oxidation in proportion to the amount of hydrogen peroxide that we add. But you see that we need to add quite large amounts of hydrogen peroxide to induce oxidation of the glutathione pool, and that this response is not particularly linear. When we add GSSG directly to these cells, we see nothing at all. GSSG cannot easily pass or cross the plasma membrane of yeast. But now when we overexpress OPT1 in these cells, we see that the response to H2O2 is unchanged. But when we titrate now in GSSG, we see that we have this very nice linear increase in roGFP GRX oxidation in dependence of the GSSG concentration that we supplement to the assay buffer. So what we're trying to say is that OPT1 overexpression allows for a more in vitro-like enzyme characterization, whilst we still, of course, retain the advantages of being able to screen in high throughput with these roGFP fusions in yeast. And as I said, these approaches are not just limited to glutaredoxins. What we've started to do recently is to look at other proteins, for example, protein disulfide isomerase, PDI1, and use these assays to look at the glutathione interaction with PDI, which is still not that well characterized. We don't really have a complete understanding of how reduced and oxidized glutathione are able to oxidize and reduce human PDI, and what the relevance of this is for the cell. And so now we're using these kind of approaches to characterize a whole range, actually, of other thyroidoxine superfamily proteins. Okay, I wanted to use the last 5 minutes or so of my talk to introduce a different assay that we've been using to monitor redox enzyme function. And this assay is a growth assay based upon the ability of a particular redox enzyme to rescue the viability of a strain in which both cytosolic glutaredoxins and both cytosolic thyroidoxins are deleted. So normally this strain is completely inviable. But we can rescue this quadruple mutant by expressing any of the 4 thyroidoxins or glutaredoxins in isolation. And we can do this by putting one of these glutaredoxins or thyroidoxins on a Uro3 marked plasmid, and then we can introduce our protein of interest on a second loose, for example, a Lu2 marked plasmid. And then we can use a negative selection assay by growing on 5-fluoroerotic acid, which is lethal in the presence of Uro3, to counter-select against the Uro3 plasmid. These cells, of course, can then only grow if our gene of interest is able to rescue the growth of this strain. In other words, if our gene of interest is able to take over the function of one of these glutaredoxins or thyroidoxins that's normally present in yeast. And actually what we were first interested in using this assay for was to look at the difference between a monothyl redox-active glutaredoxin and a dithyl redox-active glutaredoxin. So there's still some discussion in the literature about whether you need a monothyl mechanism or a dithyl mechanism for reduction of protein disulfide bonds. And we thought we could nicely use this approach to test whether a monothyl glutaredoxin is able to rescue the viability of a yeast strain deleted for both its thyroidoxins and both its cytosolic dithyl glutaredoxins. And so what we did is to express in this yeast strain, in this quadruple mutant, either a GRX1 or GRX2 as dithyl glutaredoxin controls, the redox inactive human GRX5, or the monothyl redox active glutaredoxin GRX7. You see that these strains all nicely grow on Hartwell's complete media, but when we supplement this media with 5-fluoroerotic acid, we see that the empty vector containing cells cannot grow. The dithyl glutaredoxin cells grow nicely. We know one glutaredoxin is sufficient to rescue these strains. the inactive human GRX5 containing cells are not able to grow, but interestingly, surprisingly, the monothyl GRX7 containing cells were nicely able to grow on these plates. And we also saw the same thing when we performed growth curves in liquid media. We saw that both dithyl and redox-active monothyl glutaredoxins can both nicely rescue the growth of this quadruple mutant strain. The same is also true of an artificial monothyl GRX mutant. So we took GRX2 here, we deleted the N-terminal, the C-terminal, or both redox site active cysteine residues. And again, that you can see that when we delete here the C-terminal redox site active cysteine to create a monothyl GRX2, this is nicely able to rescue the growth, both on plates containing 5-FOA and in liquid media containing 5-FOA. We can also use this assay to ask whether monothyl and dithyl glutaredoxins can reduce other enzymes in the cell. So for example, by growing on media lacking methionine, we can ask whether these GRXs are able to reduce PAPS reductase. And we find, consistent with published literature, that actually neither dithyl nor monothyl glutaredoxins in yeast are able to reduce PAPS reductase. None of these cells can grow in the absence of methionine. We can also test whether they can reduce methionine sulfoxide reductases by using media lacking methionine but supplemented with methionine sulfoxide now as a source of methionine. And these cells will only grow if the glutaredoxin that we introduce to them is able to reduce methionine sulfoxide. And we see here no difference at all in the growth. So they can all grow, and we see no difference at all in the growth dependent upon whether we express a dithyl glutaredoxin such as GRX1 or GRX2, or a monothyl glutaredoxin such as GRX7 or the cysteine mutant of GRX2. And finally, using roGFP fusions, we also see no difference in the response of a roGFP GRX2 fusion, yeast GRX2 here, compared to a roGFP2 GRX7 fusion. We see both oxidation and subsequent recovery kinetics are essentially indistinguishable. So the conclusion that we have to take from this, I think, is that monothyl glutaredoxins can reduce a variety of different protein disulfide bonds. And therefore we can say that a dithyl mechanism is not required per se for protein disulfide reduction. We can also use these yeast growth assays to, of course, screen a whole bunch of other proteins of interest. So I just wanted to show you two examples. Here you see looking at the ability of thyroidoxines to rescue the growth of these yeast strains. And you see that while normal wild-type thyroidoxin 1 or 2 can nicely rescue the growth of this quadruple mutant, when we make a monothyl thyroidoxin, this, in contrast to glutaredoxins, is completely unable to rescue the growth, as is the completely cysteineless thyroidoxin. We can also screen, for example, other proteins like pdis, and we find that in the case of the yeast pdis, these cannot rescue the growth of this quadruple yeast mutant. We were also interested in looking at human pdis as part of our ongoing interest in human pdis. And in contrast to the yeast pdis, we see that all different types of mutants of human pdi that we make can nicely rescue the growth of this quadruple mutant. So it seems that at least human PDI1 can take over the function of the thyroidoxins or glutaredoxins in the yeast cytosol and can restore the viability of a strain deleted for all of these proteins, which is quite interesting. And we're trying to understand this further now. In contrast, when we look at a class 3 type glutaredoxin, which we've been trying to make into a redox active glutaredoxin, we find that we are not yet, or we have not yet been able to activate them sufficiently such that they can rescue the growth of one of these quadruple mutant yeast strains. Okay, so just to finish off, I hope that we've been able to convince you that we've developed a series of new in vivo assays that allow the semi-quantitative and high-throughput screening of thyroidoxin superfamily enzymes. And these assays, of course, include monitoring kinetics of roGFP oxidation with hydrogen peroxide, and likewise by monitoring kinetics of oxidation with GSSG using OPT1 overexpression. and also by looking at the ability of these proteins to rescue the viability of a quadruple mutant yeast strain deleted for endogenous thyroidoxins and glutaredoxins. I hope that we could show that our assays enable the rapid screening of multiple mutants without the need for protein purification and in vitro characterization. And I think also what might turn out to be more interesting in the future particularly is that our assays allow the enzymes to be monitored in a native-like cellular environment. And we could perhaps, for example, look at things like post-translational modifications, which are much more difficult, of course, to analyze in vitro. And as I've mentioned already, this technique is applicable to other redox enzymes, for example, peroxyredoxins and glutathione peroxidases, as we've shown previously. So just to finish, to acknowledge the people involved in this work. So here, Yannick Zimmerman is a PhD student in my lab. He has been involved with the vast majority of this work. Our collaboration partners, Marcel de Ponte is our protein biochemist specialist who we collaborate on all of these projects with. Also to Jan Riemer in Cologne, to these people, to funding, and thank you for your attention.